Sources, intake, and accumulation of depleted uranium in farm animals. Depleted uranium primarily enters the environment through military activities, particularly from the use of depleted uranium munition. This results in contamination of soil and water, which can subsequently affect agricultural areas. Depleted uranium can accumulate in soil. The average concentration of uranium in natural soil is about 2 parts per million, but higher concentrations can be found near phosphate deposits or military sites. Farm animals can ingest depleted uranium through contaminated grass, soil, or water. Direct ingestion occurs when animals consume soil particles or plants that have absorbed uranium from the soil. It can also be inhaled as dust particles, which may settle on pastures and crops, leading to further contamination. The accumulation of depleted uranium in farm animals varies based on several factors. Young animals tend to absorb more uranium compared to adults when exposed to contaminated feed. This is due to their developing physiological systems. Different species may also exhibit varying levels of absorption and retention. Soluble forms of uranium are more toxic and are absorbed more efficiently than insoluble forms. For example, soluble uranium compounds can lead to significant renal toxicity at lower exposure levels compared to insoluble compounds. Once ingested, uranium is primarily eliminated through urine. However, it can accumulate in organs such as the kidneys and bones, where it may remain for extended periods after exposure has ceased. Depleted uranium consumed through feed or water is absorbed in the gastrointestinal tract, with a portion entering the bloodstream and depositing in various tissues. Inhaled DU particles can lodge in the lungs, with some particles dissolving and entering the bloodstream. Thermal absorption is a less common route, but could occur if animals have direct contact with DU-contaminated dust. Bones are the primary sites of DU accumulation due to its chemical affinity for calcium. The kidneys are critical sites for DU excretion, but also for accumulation due to its nephrotoxic properties. Depending on exposure levels, DU can accumulate in the liver and other soft tissues. Our current understanding of uranium's potential neurodevelopmental effects is based entirely on animal studies. Research indicates that uranium accumulation in brain tissues can lead to behavioral changes and cognitive impairment. Animals exposed to uranium dust have exhibited alterations in activity levels and anxiety-related behaviors due to the accumulation of uranium in critical brain regions. This can affect their survival and overall well-being. In rodents, exposure to DU during development actually accelerates the appearance of a number of behaviors such as a writing reflex, forearm placing, grasping, swimming, weight gain. Adult animals exposed to DU during development also performed worse on a test of working memory and had smaller brain weights, expressed as a percentage of body weight. Urinary excretion of depleted uranium in humans has also been linked to high serum prolactin levels and neurocognitive performance. Uranium is known for its nephrotoxic properties, which can lead to severe kidney damage in animals. Chronic exposure can lead to increased incidences of chronic kidney disease among the affected populations. Uranium has been identified as a reproductive toxicant in various animal models. Exposure can lead to fertility problems, developmental issues in offspring, and potential teratogenic effects. The endocrine disrupting nature of uranium further complicates its impact on reproductive health, increasing risks of reproductive cancers in the exposed animals. Uranium exposure can compromise the immune system, making animals more susceptible to infections and diseases. The presence of uranium in the body has been linked to changes in immune cell count and function. In severe cases, high levels of uranium exposure can lead to increased mortality rates among livestock due to the compounded effects of organ damage and systemic toxicity. Uranium exposure can have different effects on ruminants, such as cattle and sheep, compared to non-ruminants, such as pigs and poultry. These differences are largely due to variation in digestive physiology, metabolism, and the mechanics of uranium absorption and toxicity. Ruminants have a complex stomach structure that includes multiple compartments, rumen being one of them. This allows for fermentation and microbial digestion, which can influence how uranium is metabolized. The presence of specific microorganisms in the rumen may alter the bioavailability of uranium compounds, potentially affecting its absorption and toxicity. Non-ruminants possess a simpler digestive system with a single-chambered stomach. This difference can lead to more straightforward absorption of uranium from the gastrointestinal tract, potentially resulting in higher systemic exposure levels for non-ruminants when consuming contaminated feed or water. The fermentation process in ruminants may lead to slower absorption rates of uranium. Additionally, certain soluble uranium compounds may be more effectively detoxified by microbial action in the rumen before entering systemic circulation. Non-ruminants may absorb uranium more rapidly due to their digestive physiology, leading to quicker onset of symptoms following exposure. The absence of fermentation processes means that non-ruminants might experience more acute toxic effects from soluble uranium compounds. Research indicates that ruminants may exhibit specific renal responses to uranium exposure, with the potential for different thresholds of toxicity based on their unique metabolic pathways. Renal damage can still occur, but may be influenced by factors such as diet and microbial activity. Non-ruminants tend to show more immediate renal effects following exposure to uranium, particularly from soluble compounds. The kidneys are a primary target for uranium toxicity across species, but non-ruminants may experience quicker manifestations of renal dysfunction. Behavioral changes in ruminants exposed to uranium may be less pronounced initially due to their societal structures and coping mechanisms. However, chronic exposure can lead to significant changes in behavior over time. Non-ruminant species often display more rapid behavioral changes following exposure, including lethargy or reduced feeding behavior. These immediate responses can impact overall productivity and health. Both ruminants and non-ruminants can experience long-term health effects from uranium exposure, including reproductive issues and chronic kidney damage. However, the specific manifestations and severity may differ based on species-specific physiological traits. Animals exposed to uranium may experience stunted growth due to its toxic effects on metabolism and nutrient absorption. Uranium accumulation can lead to decreased feed efficiency, where animals fail to convert feed into body mass effectively, resulting in lower weight gain compared to unexposed counterparts. Uranium exposure has been linked to reproductive toxicity, leading to decreased fertility rates and complications during gestation. For instance, animals may experience reduced litter sizes or increased rates of fetal loss, which directly affects overall productivity in breeding operations. Uranium can also cause developmental abnormalities in offspring, impacting their viability and growth potential, which can have long-term implications for livestock productivity. In dairy cows, uranium exposure can lead to lower milk production levels. The stress and health impacts associated with uranium toxicity may reduce lactation performance, affecting both the quantity and quality of milk produced. Uranium contamination can compromise the immune system of farm animals, making them more susceptible to infections and diseases. This increased vulnerability can lead to higher veterinary costs and reduced productivity due to illness or treatment downtime. Long-term exposure can result in chronic health problems that further diminish productivity through reduced feed intake, poor body condition, and increased mortality rates among livestock. Uranium exposure may lead to behavioral changes in animals, such as increased anxiety or decreased activity levels. These changes can affect feeding behavior and social interactions within herds, potentially leading to lower overall productivity. Animals exposed to uranium may exhibit signs of kidney damage, which can manifest as abdominal swelling or distension due to fluid accumulation. This is particularly evident in cases of acute renal failure. Alterations in urination patterns, such as increased urine volume or changes in color, can indicate renal stress or damage. Inhalation of uranium particles can lead to respiratory distress, characterized by labored or rapid breathing. This may be accompanied by coughing or nasal discharge. Long-term exposure may result in chronic respiratory conditions, which can be observed through decreased activity levels and reluctance to move or exercise. Animals may become lethargic or less active due to discomfort or pain associated with uranium exposure. This behavioral change can be a visible indicator of underlying health issues. Increased anxiety or stress responses can lead to changes in social behavior, such as isolation from the herd or aggressive interactions with other animals. Young animals exposed to uranium may show signs of stunted growth or delayed development compared to their peers. This can be visually assessed through size discrepancies. In severe cases, particularly with prenatal exposure, physical deformities may occur in offspring, affecting their viability and health. Uranium exposure leads to a decline in overall health, which may manifest as a dull, rough coat or skin lesions indicative of systemic illness. Irritation or dermatitis may develop due to direct contact with contaminated environment or materials. In the cases of acute exposure, symptoms can develop within a few days to weeks after exposure to uranium aerosols, particularly if the uranium compounds are soluble. For instance, renal effects have been observed in laboratory animals exposed to soluble uranium compounds at concentrations as low as 0.13 mg of uranium per cubic meter over intermediate durations. Acute symptoms may also arise from ingestion of contaminated feed or water. Changes in urine output are signs of renal dysfunction. They can occur within days following significant oral exposure to uranium compounds. For chronic exposure scenarios, such as continuous ingestion of low levels of uranium over time, symptoms may take longer to develop. Renal damage and other health effects may not be immediately visible but can become apparent over weeks or months of consistent exposure. Early signs, such as changes in behavior, reduced activity levels, and mild gastrointestinal disturbances, may appear within a few days after initial exposure. More pronounced renal symptoms, such as altered urination patterns or abdominal swelling, may become noticeable within a week or two following significant exposure. Both milk and meat can contain measurable levels of uranium, raising concerns about food safety and public health. Studies have indicated that uranium can accumulate in the tissues of livestock, including muscle and fat. The activity concentrations of uranium isotopes in meat products have been reported to range from 1 millibecquerel per kilogram to 20 millibecquerel per kilogram, with U-235 levels being significantly lower than those of U-238. Milk from lactating animals can also contain uranium, reflecting the animal's exposure levels. Activity concentrations in milk products around the world are typically found between 0.1 millibecquerel per kilogram and 17 millibecquerel per kilogram. This accumulation may pose risk to human consumers, especially if consumed regularly over time. The conversion of food systems plays a crucial role in the availability and potential health risks associated with depleted uranium. Understanding how depleted uranium enters the agricultural supply chain, from soil and water contamination to plant uptake and animal consumption, it is vital for ensuring food safety. Monitoring and regulatory measures are essential to manage these risks effectively, protecting both animal health and consumer safety. The supply chain involves multiple steps: farming, harvesting, packaging, transport, distribution, and retail. Each stage presents an additional opportunity for contamination if proper safety measures are not followed. Awareness of contamination sources is crucial for consumers who might be at risk due to dietary choices involving animal products from contaminated regions. Thank you for listening.